It is a privilege and an honor to introduce a scholar of such outstanding rank. Dr. Dembski has a long and impressive intellectual pedigree. He is a graduate of the University of Illinois at Chicago, where he earned a BA in psychology, an MS in statistics, and a PhD in philosophy. This gifted intellectual also received a doctorate in mathematics from the University of Chicago in 1988 and a Master of Divinity degree from Princeton Theological Seminary in 1996. Beyond this, William Dembski has held National Science Foundation graduate and postdoctoral fellowships. Suggesting something of the breadth of his interest, Dr. Dembski works quite comfortably in the fields of mathematics, philosophy, and theology. Who among us can say that? But two of the main reasons why Dr. Dembski warrants our attention this morning is, first of all, because he is a fair-minded critical thinker. His writings demonstrate the characteristics or traits that make up such reasoning. Having read many of Dr. Dembski's works, I can accurately affirm that he evidences intellectual courage in abundance. Indeed, he has repeatedly demonstrated the willingness to face and assess fairly ideas, beliefs, and viewpoints to which we have not given a serious hearing in our politically correct culture. Moreover, Professor Dembski reveals intellectual good faith in that he holds himself to the same rigorous standards of evidence and proof to which he holds his critics. And these two traits, or intellectual habits, readily flow into a third, namely intellectual perseverance. That is, Dr. Dembski has shown a willingness to pursue intellectual insights and truths despite difficulties, obstacles, and frustrations. Indeed, he has been called all sorts of names by his vituperative and mean-spirited critics, and yet he calmly continues the important intellectual work that must be done. Second, and finally, I commend to you Dr. Dembski this morning because he is nothing less than a prophet for our age. No, he doesn't run off into the desert on occasion eating locusts and wild honey, but he does speak the truth in love to an age that has grown weary of truth, to an age that is all too comfortable and self-satisfied with the worn-out and easy answers of yesteryear. More important, Dr. Dembski's worldview, which is inclusive, not exclusive, in that worldview, there is room enough for both science and faith, for the life of the mind and the life of the soul, for intellectual truth and vital piety. And so for all these reasons and more, I hope that you will give Dr. Dembski a very warm welcome as he comes to the pulpit.
Thank you very much. That was extremely gracious. So, with that, I'm going to try to coordinate a PowerPoint that I'm looking at here with one that's going on there. So we'll try to keep this all synchronized. And let's see, do we? Yes, we are. We are looking at things up there. Well, yesterday I gave an overview of as it were, the theological import of intelligent design, and then in the evening I did a colloquium on some more technical aspects of the work. And so today I want to focus on what's going on in the biology? Why is design an issue again? And show you some pretty pictures and a video that's going to knock your socks off. So let's see if this gets coordinated. This and this. Okay, so my title is provocative, trying to draw out a crowd on a Wednesday morning. And so here, what I'm keying off of is a claim in Richard Dawkins' The Blind Watchmaker. Although atheism might have been logically tenable before Darwin, Darwin made it possible to be an intellectually fulfilled atheist. And so what I want to examine is whether that's actually the case, whether you can be an intellectually fulfilled atheist on the basis of Darwin, or just an atheist but without the intellectual fulfillment. So first off, I think we need to be clear what we're talking about. And here, you will not be able to read this, but I will zoom in on it. I think that this will make the point of what's really at stake. I received an invitation back in 2001 to take part in a debate. And let's zoom in a little bit further. This is from the American Museum of Natural History, downtown New York, Manhattan. If you will, the belly of the beast for evolutionary thinking. The email I got said, "I would like to invite you, Dr. Dembski, to participate in an upcoming panel discussion at the museum, Blind Evolution or Intelligent Design." A debate on evolution. I just want to make sure that that we are synchronized. And now the thing is, debates have debate questions, debate topics, so that was to be the question. Now when I showed up at the debate, there was an auditorium and there were program guides which were passed out. This was actually the title on the program guide. Now do you notice the difference? Let's see if we can back up. You know, we go here and we go here. Well, suddenly the word blind had been omitted, okay? Now, intelligent design, although it's compatible with special creationism, God just boom, 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 things just, as it were appear presto changeo, it is also compatible with a form of evolution in which God works the design through an evolutionary process, and that design then is empirically evident. So this is really a question of implementation. How do you get the design in there, whether through an evolutionary process, through some sort of special creative process? Those are questions downstream, as it were, for intelligent design. But the real question is whether there's intelligence involved and whether that intelligence is discernible. But by posing the debate question this way, evolution or intelligent design, that gets really fogged over. Now the thing is, I don't think the people who wrote these programs meant to be dishonest, because I think for the vast majority of people, evolution means, Darwinian evolution means a blind, purposeless form of evolution. And so let me just indicate to you that really is the widespread view. Stuart Kauffman, who is not a proponent of this evolutionary, of the Darwinian evolutionary view. He's an outsider. He's not a design person either. He's a self-organizational theorist. Writes the following in his book, At Home in the Universe. He says, biologists now tend to believe profoundly that natural selection is the invisible hand that crafts well-wrought forms. It may be an overstatement to claim that biologists view selection as the sole source of order in biology, but not by much. If current biology has a central canon, you have now heard it. So natural selection, that was Darwin's key notion. Michael Shermer, a skeptic, published a Skeptic magazine, also has a monthly column in Scientific American. No one, and I mean no one, working in the field is debating whether natural selection is the driving force behind evolution, much less whether evolution happened or not. Jerry Coyne, an evolutionary geneticist at the University of Chicago, writes, there is only one going theory of evolution, and it is this: organisms evolve gradually over time and split into different species, and the main engine of evolutionary change was natural selection. And then he goes on from there. So what we might say is that Darwin offered a promissory note that this mechanism of natural selection and random variation could account for all of biological information. I'm using biological information loosely, not as technically as I did last night in my talk, but just that matter has to be configured in very particular ways if it is going to be alive. Okay, and and natural selection and random variation are supposed to be the means by which this happens. Now, let me just say a little bit about natural selection and random variation. These are non-teleological notions. That means there's no goal or end-directedness to them. The neo-Darwinian view of evolution, which is the one that gets taught in all the textbook, basically sees variation as coming about through random genetic errors. Okay, so it's like you can think of your DNA as really a typescript, and it's just as you're typing or copying something, you make a mistake. Well, that's what happens. There are error rates by when DNA gets copied, and it's those errors. Some of those might be beneficial, and then they would get selected by natural selection. But the thing is, those errors, they're not correlated with any future benefit to the organism. The organism isn't thinking, or the evolutionary process isn't thinking, gee, I'd like to mutate a few things, and down the line I'll get a few extra enzymes and proteins, and down the line I'll get some new cells, and then I can form some nerve cells, and then maybe I can get an eyeball out of it. That's not the way Darwinian evolution works. It's very much trial and error tinkering, immediate gratification. There's an error that takes place, and then natural selection asks, can I exploit this right now for the benefit of the organism, benefit being cashed out as survival and reproduction. It's cold, hard demographics. That's all evolution is concerned about. Will this help me to survive and reproduce now? Not will this provide a benefit to my offspring 500 generations from now. So it's thoroughly non-teleological. There's no goal there, and you cannot see the divine handiwork in that process. You might want to say, well, God set it all up. Okay, that's a move people make. Those people are called theistic evolutionists. But in the process itself, there's no place that you see God's handiwork. And so Francisco Ayala, a former Catholic priest, one of the most prominent evolutionists this side of the Atlantic, has said that Darwin, his greatest achievement was to show that the creative organization of living things could be brought about without the need for a designer or creator. Okay? It's not that a designer or creator doesn't exist. It's not that this theory proves that there is no God. But if it holds, then in a sense there need be no God. Okay? If there's a God, in a sense that this God is working behind the scenes, he's a master of stealth who has set things up so that this process works itself out. And it's a brutal process, by the way. I mean, what you're talking about is then ages and ages of brutal competition that has led to us. And I remember, actually this was in 6th grade, I remember being taught by a teacher that I had quite a respect for at the time, saying that why are we here? What brought us about? Because we are the best killers. I mean, I think that in a sense, captures as it were, the logical outworking of Darwinism. Not that that's where most Darwinists take it. I think they try to soft pedal some of the brutality of the theory, but I think that's there. Okay, so let's move on. So that's, as it were, the promissory note, that this mechanism could do all of that, and I think given what I just said, you can see why somebody like Richard Dawkins would say, bully for Darwin, it makes it possible for me to be an intellectually fulfilled atheist. Well, I hope at the end of this talk that you'll see that Dawkins can continue being an atheist, but he has really no rights to claim intellectual fulfillment. So, continuing on, what would it mean to default on the note? Yep. Okay. So, Darwin proposed a test for his theory. The problem is it's an impossible test. Let me read it. This is in his Origin of Species. He says, if it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down, but I can find out no such case. Now, what he's talking about here is that his process of evolution has to work gradually. It's got to be built up bit by bit. You can't just have things magically materialize. Okay? I mean, if you're going to have things magically materialize, then you've got either a God theory or a brute chance theory. And there had been brute chance evolutionary theories, but that wasn't good enough for Darwin. He wanted to explain getting lucky is not a good explanation in science. Okay? So you want something better than that. And Darwin thought that he did have something better than that. But the thing is, this doesn't really go far enough. I mean, if we're just talking numerous successive slight modifications, I can imagine a motor being built into a motorcycle by adding molecules bit by bit. It gradually evolves, as it were, but it would be a design process. It would not be a Darwinian process. So I think we need to look at this test a little bit more closely. Also, this claim could not possibly, that's way too strong for most of science. Could not possibly, that's the level of mathematics. 2 plus 2 could not possibly equal 5. Okay? But the Earth could not possibly be the center of the universe. Well, God could have created it that way. It's just that God didn't create it that way. So most scientific claims are not at the level of could not possibly. This is too strong. Most scientific claims are at the level of probabilities rather than impossibility. And then also this claim about absolutely breaking down. Most scientific theories don't absolutely break down. You can always come up with some quick and dirty ad hoc fix. You just add some auxiliary hypothesis. Theologians know all about this, right? So scientists do the same thing. And then again, numerous successive slight modifications. It's not just that this has to be gradual, but it has to follow this Darwinian mechanism of natural selection and random variation. But anyway, I think to visualize what's going on here, and essentially what Darwin is saying that if you're going to get to this island, this new biological form, and I'm making an analogy, a visual analogy, you're starting here, you've got to be able to take little hops, little baby steps, as it were, to get there. On the other hand, this wouldn't be good, okay? You couldn't hop that whole thing. Okay, so that's the intuition there. Now, I think you need to reformulate that test, though. And I think Darwin was trying to be honest. I think he was trying to, but I think he just wasn't very careful in formulating it. So I think if I were to restate his challenge, it would be as follows. If it could be demonstrated that any complex biological system, instead of organ, let's talk about complex biological systems, that's more general, existed that is highly unlikely, as opposed to could not possibly, highly unlikely to have been formed by a Darwinian selection mechanism, variation selection mechanism, acting over numerous successive slight modifications. It's not that some designer, atom by atom, constructed a motorcycle from a motor, but that that it happened by Darwinian trial and error tinkering process. Darwin's theory would break down, not absolutely break down, but break down. It would be a challenge to it. Okay, so I think this is a more reasonable test. So the question then is, well, where might we look for that more reasonable test? What we're going to have to do, this is going to be, as it were, a case-by-case test. And we're going to have to find some complex biological system and show that the test breaks down. It's not the burden of the person challenging Darwin's theory to show that every complex biological system satisfies this test. It's not enough to find that there is some that would do it. So what should we be looking for? Well, in Darwin's day, Darwin didn't know any molecular biology. When he looked at the cell, he basically saw it as a little blob of jello enclosed by a membrane. It was extremely simple. In fact, he thought that cells could spontaneously generate. The science at that point in the 1850s, 1860s had come to the place where they said, okay, mice and flies, they can't magically come about on spontaneously generate on rotting meat or in rags, but single-cell organisms, they thought, could. Okay, but now, given the revolution in molecular biology, electron microscopy, what we know about the cell, there's a world of tremendous complexity there that Darwin did not suspect, and which, I'd put it to you, offers precisely those complex systems that Darwin thought did not exist and would not withstand his theory. So what are we looking at? Well, what sorts of complex biological systems might test this theory? And a friend of mine, Michael Behe, has written a book called Darwin's Black Box. It's about 10 years old now, in which he identified certain systems and a particular feature of those systems which would throw a real monkey wrench into Darwinian explanations. Now, what was he finding in these systems? Still going on. Well, what he found inside the cell was, if you will, a serialistic Lilliputian realm. You have within the cell self-replicating robotic manufacturing machines, information processing, storage, and retrieval, signal transduction circuitry, high-efficiency, high-tech nano-engineered motors, transportation and distribution systems, automated parcel addressing, UPS labels or zip codes. I mean, in a sense, you've got everything in the cell is very tightly choreographed. You know, I think we often have the image that the cell is just this floating business of blobs, and in fact, things have to be targeted to very specific locations, particular proteins, particular enzymes. Okay, so I want now to show you a video which I think will really nail this down for you.
In the years since Kenyon's rejection of chemical evolution, science has revealed the details of an entire system of information processing that bears the hallmarks of intelligent design. With computer animation, we can enter the cell to view this remarkable system at work. After entering the heart of the cell, we see the tightly wound strands of DNA, storehouses for the instructions necessary to build every protein in an organism. In a process known as transcription, a molecular machine first unwinds a section of the DNA helix to expose the genetic instructions needed to assemble a specific protein molecule. Another machine then copies these instructions to form a molecule known as messenger RNA. When transcription is complete, the slender RNA strand carries the genetic information through the nuclear pore complex, the gatekeeper for traffic in and out of the cell nucleus. The messenger RNA strand is directed to a two-part molecular factory called a ribosome. After attaching itself securely, the process of translation begins. Inside the ribosome, a molecular assembly line builds a specifically sequenced chain of amino acids. These amino acids are transported from other parts of the cell and then linked into chains often hundreds of units long. Their sequential arrangement determines the type of protein manufactured. When the chain is finished, it is moved from the ribosome to a barrel-shaped machine that helps fold it into the precise shape critical to its function. After the chain is folded into a protein, it is then released and shepherded by another molecular machine to the exact location where it is needed.
This is absolutely mind-boggling to perceive at this scale of size such a finely tuned apparatus, a device that bears the marks of intelligent design and manufacture. And we have the details of an immensely complex molecular realm of genetic information processing. And it's exactly this new realm of molecular genetics where we see the most compelling evidence of design on the Earth.
This is state-of-the-art animation, and I think it gives you just some insight into the handiwork of the Creator, and I think this is just the surface, because I mean we're just barely understanding these systems even now. I think if you go back into the 60s, and there were visions or movies about what's inside the cell or inside the body. There's a lot of blobs floating about. This isn't blobs. This is all tightly orchestrated. This is an automated city where everything has a purpose and everything is doing something important. Okay, well, let's look at that a little bit more closely. What are some of these systems then that are inside the cell? And one that's gotten really the most play, it's become, as it were, the icon of intelligent design, is a little bidirectional motor-driven propeller, which resides, if you will, on the backs of certain bacteria. E. coli is the one that's been studied the most. And this thing can spin at up to, I have 17,000 RPM, but they've been known to spin it up to 100,000 RPM. It's basically a little propeller that propels the bacterium through its watery environment. It's bidirectional, so if it's spinning at 100,000 RPM, it can actually stop in a quarter turn. It's spinning full speed in the opposite direction. So imagine if you had a blender that could do that, and what you might be able to blend. It would be amazing. But anyway, this thing can do the job, and it's bidirectional because you have a bacterium. It's trying to swim up a food gradient. So it's in this watery environment. So, there's some food there. It's trying to swim up, but it's so small that water is in constant motion. There's Brownian motion, this random jiggling of water molecules, and so it's throwing the bacterium off its course. And when it gets off sufficiently, then it changes direction, tumbles, and tries to reset itself and go back up this food gradient. So it's basically doing what mathematicians would call a random walk with drift. It's trying to drift up this food gradient. It's a marvelous device. In order to do that, by the way, this is just the motor part and the propeller. There has to be a whole signal transduction circuitry where it's taking in information from the environment, telling it where the food is, where the concentrations are. But just a marvelous device. Howard Berg at Harvard will call this the most efficient machine in the universe. It requires about 40 protein parts, and you look at this, you need to be an engineer to understand this system, at least in detail. Broadly we can understand it. You've got this long whip-like tail. That's actually the bulk of the flagellum. You have a hook that's a universal joint, a drive shaft, various discs that mount this on the cell membrane, stators, O-rings, and then you've got an acid-powered drive that moves this thing. Let's see, I've got still another one, and here you have I'm showing you the signal transduction circuitry. So you have these chemical receptors which are feeding then in to the flagellum and telling it how to spin. Okay. So the question is, well, how did something like this arise? Okay. Now, certain things to be aware of with systems like this. They're multi-part, lots of things going on there. They're functionally integrated. Every part is doing something significant in this. You can't just eliminate them willy-nilly and still maintain function. They're non-simplifiable, at least broadly. If you're going to have a propeller that's propelling something through a watery environment, you're going to have a propeller, something that's outside the bacterium. It's got to be able to get through the cell membrane as it's built. You've got to have some sort of drive shaft, a motor, something that mounts this apparatus onto the cell membrane. So normally the bacterial flagellum that's been most studied is the one in E. coli. There have been some simplifications in the sense that you can maybe eliminate a few of the discs in mounting it, but conceptually, there are all these things which have to be there, and you really can't simplify it and maintain that function. And further, there's no hidden structure. Okay, and I think this is really important. This is why he titled his book Darwin's Black Box. It's not like we're dealing here at the biochemical level. It's not that we can go to some lower level where there's stuff going on that we have no clue about which might be leading to it. So it's not like if we go to some smaller level of resolution, there might be little green men that are building these things, okay? That's not going on. You go below this biochemical level, and you're basically in the level of inorganic, or just brute chemistry, And so you don't have anything that's biologically significant if you go below that level, okay? In a sense, I think Darwin really hid behind the complexities of life. I mean, he looked at the eye and said, yeah, it could evolve. Well, the eye is just incredibly complicated. It's got so many millions, billions of cells. In a sense, the Darwinist says, well, I can imagine how a light-sensitive spot could evolve into an eye. Prove me wrong. Show me that it didn't happen that way. But with systems like this, I mean, these are inside a given cell. They're much simpler. They're still quite complex, but much simpler, and we can get a handle on them in a much better way to determine what their design characteristics are and what would be involved in their evolution. Okay, so when we have systems like this, they're called irreducibly complex. Bacterial flagellum is one such system. So question then is, how does evolutionary theory attempt to explain something like the bacterial flagellum? Now, I think often when we think of evolution, a Darwinian form of evolution bringing something about, we think of it taking something and, as it were, enhancing it. You take a given function, you just make it better and better. So if you were to explain how the eye evolved, you might say, well, it started as a light-sensitive spot, then it cupped a bit so that because it's cupping, it could tell a direction source for the light. Then it formed an enclosure with a little pinhole, which served as a pinhole camera. Then it formed a lens. I mean, all this is hand-waving, by the way, because eyes have to be built in embryological development. There've got to be genetic changes. None of this is ever articulated by the Darwinists. These are what critics will call just so stories, arguments from imagination, if you will. But arguments from imagination actually go much further in the scientific world than anything that might implicate a deity. So God is unthinkable. Arguments from imagination, those are still acceptable, which I think is an indication of how far we've come. But so how do they explain this? Well, there's one preferred mode of explaining these things, which I would call coevolution and co-option. Co-option, what is that? Imagine that you're in prison. You've got a laptop, you've got a radio and a microwave. And you happen to be an electronic genius. You want to watch the Super Bowl, and so you put those things together and you form a working television. Okay, so you cannibalize various parts from this and put it all together. That's co-option. You take things that were targeted for different uses and put them together for a new use. And so coevolution and co-option, basically what it means is that evolution works by borrowing. It has to take existing things. It does not just generate things from scratch. So it takes things, reworks them, reshuffles them, and so the idea is that you can bring about a bacterial flagellum by basically, you start with some primitive or precursor structure that was doing something else. And then you modify it, and then maybe it's doing something else again. You modify it again, it's doing something else. So the structure and the function are both undergoing change. Okay, and eventually you can bring about the flagellum. So you might imagine this. If you were to explain the evolution of a mousetrap by coevolution, co-option scenario, you might say, well, the mousetrap did not really start as a mousetrap. It started as a doorstop. There was this base that was a doorstop. And then it became a tie clip because there was this spring and holding bar, and so basically it served as a tie clip. But it still could not catch mice there until we added a catch and a holding bar, and catch and what is it? The holding bar. And so it's the hammer and the spring that forms the tie clip, and then the catch and the holding bar, and then we get a full-fledged mousetrap. So you kind of see its structures, the structure changes, and the function changes. Okay, so that's the argument. And what's the evidence for this? I mean, it might be, it might not, right? I mean, it could go either way. Well, what biologists have found is that inside the bacterial flagellum, there is embedded, as it were, something called a type 3 secretory system. This is a little pump. It requires about 12 protein parts instead of 40. This was the poison delivery system that killed about a third of the population of Europe in the 14th century. This is what Yersinia pestis, the bubonic plague bacterium, used to pump poisons in and kill people. And it's as it were embedded, the proteins for this are as it were embedded in the flagellum. And in a sense, it makes sense that something like this would be in the flagellum, because think of how the flagellum is built. It's all these proteins are inside the cell, and then you've got to get stuff outside the cell. So there's got to be some sort of secretion to get that flagellar tail outside the cell. Well, something like this is operating in the flagellum, and so this system seems to be embedded in it. Okay? So the claim then is made, well, this could have been an evolutionary precursor to the flagellum. Okay? Now, I think the problem with an argument like this is that just about any complex system that you look at will have subsystems that could do other things. I mean, I look at this motorcycle, there's a motor there, right? I mean, that motor could be doing something else, okay? But getting from a motor to a motorcycle still is a design work. And likewise, getting from a type 3 secretory system to a bacterial flagellum is going to require a lot of redesign. And it's not at all clear that a Darwinian mechanism could do that. In fact, the best evidence these days is not that the flagellum evolved into rather that the type 3 secretory system evolved into the flagellum, but that the flagellum, as it were, devolved into the type 3 secretory system. Now, water's been around a long time. Bacteria have had to get around water for billions of years. What does the type 3 secretory system do? It infects multi-celled organisms. How long have multi-celled organisms been around? Well, about 500, 600 million years. Okay, and so in fact, the best evidence is suggesting that you have as it were, evolution going from more complex to simple, and that's fine. Okay, evolution can work that way, but that means that we haven't explained the flagellum. Thing is, the whole point of evolution is to explain complexity in terms of simplicity, not simplicity in terms of complexity. So really, this explanation falls flat. I mean, it's speculative. There are no details. You don't have a fully articulated Darwinian path which would allow us to assess whether this Darwinian mechanism is actually able to do all this work. And so the way I would put it is the difficulty facing Darwinism. What's needed is a complete, fully articulated evolutionary path, and not merely a possible oasis along the way. To claim otherwise is like saying we can travel from Los Angeles to Tokyo because we've discovered the Hawaiian Islands. And throwing in some Fiji Islands and Tahiti and other things, along the way is not going to help you much either. Okay, I mean, you need a path. Right? It's got to be a continuous path, not just a few, because people have said, well, there could have been some other system, and then some others. They've pointed to a few other ones, but those type 3 secretory system, we can actually point to those. These others that they've proposed are pure speculation. I mean, they don't even exist. So I think that's the problem. But the problem, and here we see it again, but I think the problem goes even deeper. Okay. What I've just described is this irreducible complexity and the challenge it poses once we've got life. But the question, the same argument applies to the origin of life itself. The only living forms that we know are cells, which have a full DNA, RNA, protein synthesis machinery. Okay? That machinery is incredibly complex. Okay? It requires about 60 enzymes just to get into your DNA and work with it. Okay? So you've got your DNA, about 3 billion base pairs there. It's super coiled. You're going to have to get in there and uncoil it. You got to read it. You got to get that information out into mRNA. Then you've got to get the mRNA out to the ribosome. The ribosome, that's another 50 or 60 protein and RNA parts. These are extremely complicated machines. The thing is, life as we know it is impossible without that whole protein synthesis mechanism. Now, I want you to get this, because with the bacterial flagellum, you might say, well, there was this simpler system that the flagellum could have evolved into the flagellum. Okay? The thing is, that simpler system, life could still have existed with that simpler system. What's simpler than this? We don't have any example of a simpler system that would allow for life that could then evolve into this and still maintain life. So there's no simpler system that we can look to and say, oh, we could have evolved from there, because we don't have any examples of anything that's simpler that allows life to be. Okay? So that whole argument of coevolution, co-option doesn't even work when we're talking about the origin of life. And the level of complexity here just goes way beyond even what we're dealing with the flagellum. We have, as it were, layers and layers of irreducible complexity there. So I call this irreducible complexity on steroids. So we need to wrap things up. Where are we then? Has the note been defaulted on? I would say there are no, I'm quoting here James Shapiro, a molecular biologist at the University of Chicago, who reviewed Behe's book back in '96, says there are no detailed Darwinian accounts for the evolution of any fundamental biochemical or cellular system, only a variety of wishful speculations. It is remarkable that Darwinism is accepted as a satisfactory explanation for such a vast subject, evolution, with so little rigorous examination of how well its basic theses work in illuminating specific instances of biological adaptation and diversity. Now I want you to look at this first sentence. There are no detailed Darwinian accounts for the evolution of any fundamental biochemical system or cellular system. Got it? Okay, let's go to the next quote. This is 5 years later from Franklin Herald. Okay. There are presently no detailed Darwinian accounts of the evolution of any biochemical or cellular system, only a variety of wishful speculation. This is in his book, The Way of the Cell, Oxford University Press, 2001. There's no footnote here. He does not cite James Shapiro, okay, which leads me either to believe that he's plagiarizing, or this is such a self-evident truth when people start thinking about it that they come up with the same terminology to describe it. By the way, I debated Ken Miller in 2002 at the World Skeptics, I think it was a quadrennial skeptics meeting in Burbank, California. This is in front of 700 skeptics and atheists. And I pointed this out because he's saying the flagellum, no problem, it can evolve. I said, what do you do with something like this? A claim. I mean, Franklin Herald, cell biologist, Colorado State University. His response was, well, Franklin Herald's an old guy. He's been retired about 15 years. That was it. That's an argument. So it's and I've got a few others, but I need to run. But the point is, this problem is unsolved, okay, on Darwinian grounds. But what should we then say? Does this prove design? I mean, we've got design language. I mean, the tension is felt even by the atheists. Richard Dawkins' biology is the study of complicated things that give the appearance of having been designed for a purpose. Francis Crick, he was diehard atheist. Late Francis Crick, co-discoverer with Jim Watson the structure of DNA. Biologists must constantly keep in mind that what they see was not designed, but rather evolved. A similar quote by Dawkins. So what do we make of this? Well, here I want to just say that it seems that a design inference is a very natural one. If we think of design, intelligent design, as the study of patterns in nature that are best explained as the result of intelligence, then it seems that there's a design inference that readily comes to mind. Now, that design inference is not the following. Okay, this is, I think, how Darwinists would like to caricature what we're doing. They would go as follows. So how Darwinists understand the design inference. Shucks, no one has figured out how the flagellum arose by Darwinian means, okay? So gee, it must have been designed, okay? That's not what we're saying. This is, I think, a much better rendition of what we're saying. Certain biological systems have a feature, call it IC, irreducible complexity. Okay? Premise 2, Darwinian explanations have been spectacularly unsuccessful in explaining these systems. This from a theory which is supposed to be the greatest theory ever thought by Darwinists. This is supposed to, nothing in biology is supposed to make sense except in the light of the Darwinian mechanism, Darwin's theory. Okay? Premise 3, intelligent agency has the causal power to produce systems that display irreducible complexity. And we need to be engineers to understand what's going on in the cell. If we weren't engineers, we wouldn't understand how the cell is an automated city. Okay? So it's not a big stretch, therefore, to conclude, therefore, biological systems that exhibit IC, irreducible complexity, are likely to be designed. Is this slam dunk? No, but it's a good inference. Now, if we left it just there, and this is where I will leave it because we're running out of time, but I think it would still be a powerful inference. The question is, can we do science with it? And let me just I'm just going to run very quickly through testability problem. What are the proper methods for testing design? And this is where I didn't get into my work because there wouldn't have been time for it. I thought this was more important to get across to you. But a lot of my work has been on formulating information theoretic methods for identifying intelligence. What are we looking for? Specifically, what sorts of probabilistic and pattern features are we looking at which will allow us to identify intelligence? And my work in the design inference is focused on that. Functionality problem, what is a design functions design object's function? Transmission problem, what's its history? I mean, if we're right, what we're dealing with is a form of technological evolution. Implementation problem, how was the design implemented? Was it through an evolutionary process? Was it by special acts of creation or something in between? Reverse engineering, how could the design object have been come about? Perturbation, how has the original design been modified? What factors have modified it? Separation of causes problem, how does one tease apart the effects of intelligent and natural causes? That's a problem you can pose within a design framework. You can't even pose it within an evolutionary, a materialistic evolutionary framework. Think of a rusted old automobile. They're the effects of design. They're also the effects of weathering, erosion. Okay, can you tease those apart? If dealing with some art object that's been exposed, or been damaged in some way, what's the original design? What can we recover it? What's the design? What's the result of intelligence? What's the result of material processes? And they can be all mixed up. Restoration problem, how do we get back to the original design? Constraint problem, what constraints operate in when this design object functions? Information generation problem, how does the information arise? Are there layers of information? This is a neat thing. This is called the Sater repo word square. But the repo, the sower holds the wheels at work. You read it any way, this way, it's all the same, So you have this, it seems that within the DNA there are layers and layers of information. There's a whole field called digital data embedding technology, and one aspect of it is steganography, where you're basically hiding data within data. So you might have an image, and there might be a watermark, or there might be some other information that's embedded within there. And it seems that you have this sort of multi-layered embedding of information in DNA. This is high-tech stuff. This is not something that you'd expect from a trial and error, natural selection, random variation, 19th century idea that cobbles together things in a trial and error fashion. I mean, this is way beyond that. Information nesting problem, that's what I just described. Optimality problem, in what way is the design optimal? Ethical problem, is the design morally right? Aesthetic problem, is the design beautiful? Intentionality problem, what was the intention of the designer? And finally, identity problem, who or what is the designer? So you got a whole range of things that you can be working on. Lots of dissertation problems, whole research program. So with that, I will leave it. Thank you for your patience. I went over my time.